This is your first chapter in your syllabus. That is, the first chapter itself is a introduction to biotechnology. So come, let's see what and all you will be studying here. Okay. The first one is your historical perspectives and your applications of modern biotechnology and biotechnology in India and academic prospects and industrial scenarios. So first you will be studying the history, how the biotechnology in the ancient times they were been performing without any equipments or technologies. Okay, next we will be see the modern application as of now, how from ancient to modern it's evolved. We'll be seeing that, and we'll see how biotechnology in India right now, what is its progress and what all the development, and also its academic every year what and all it is taking place and how initiatively it is developing step by step, and the industrial scenario in industry, how the biotechnology has been taking place to for the new inventions or how it has been developing the biotechnology in their industrial. So that we'll be seeing in the industrial scenario. So come first, let's see your history perspectives. So can you just guess who's this scientist on the photo? He was the one termed biotechnology. Carl Erke, a Hungarian scientist who was the one who gave you the term, who gave you the name biotechnology. He was the scientist. Okay, so basically what is this biotechnology? In the sense, your biology, your chemistry and your microbiology. Okay, these three together units will give you a composed of a single term or single subject. See, in biotechnology you will be studying biology and you will applying be some chemistry and also some of your microbiology. So together these three, mixture of these three subjects, I can tell you as a biotechnology. So who was the scientist who termed biotechnology? He was Carl Erke. Okay. So now let's see what is this biotechnology. So this biotechnology itself, it gives you the two names, bio and technology. So what is this bio? Bio refers to your life, biological process, biological molecules, your living cells and everything. Okay, technology is nothing but some of the methods or process or a system, you can tell. So what is this? Basically biotechnology means you are using a biological process and performing some function or system which is useful to a mankind in your daily life. You are taking a benefit to us for the man. So from the biological process, whatever we are here for your living organism, cells, plants, everything, whatever, you are taking that and you are doing some process or method from that where you are getting benefit to us, some useful things, that you can term as biotechnology. Okay, so let me read it out a definition for you. Biotechnology, the term is a combination of two words, bio and technology, as itself I just told you. It refers to the different technology that make use of, I told you right, living cells and biological molecules to generate useful products for the benefit of a mankind. So from the biological molecules you are performing some functions. So from that you are getting a useful products for the benefit of us, in the sense a mankind. So did you get it? What is the term biotechnology? So now let's look into your next slide. So historical perspectives. So in historical, how is the biotechnology was performing? See, in history, in ancient times and all, you didn't have any microscope or DNA sequencing, polymer centrifugation, that machines, labs and all were not there. But still also in ancient times also there was performing biotechnology. So now let's have a look into that. So ancient biotechnology, when was actually biotechnology rooted? It was 10,000 years ago. 10,000 years ago in your Paleolithic era. In 10,000 years ago, which era was that? It was Paleolithic era. So now let's move on to the next slide. So I already told you in ancient times they were performing some biotechnology process, but there were no machines or equipments like that. So here is a good example for you to that explain. So you already know that you do a curds at your home using a milk. So for that, how it is possible, let's have a look into that. See, first you have to, what you will do, you will boil the milk to a certain temperature. See, every substance has a different boiling point. Okay, so first what you will be doing, a fresh milk is boiled to 100 degree Celsius and cool to 37 degree. So what you will do, so you will usually be, you don't know the degrees at home, you will be randomly heating the curd until if you have a cooker for a milk, so that will give you a whistle. Small cookers will come for the milk, will give you a whistle, you will off it, and randomly you allow it for some 1 hour depending on the climate. If it is too cold, it will just cool it in a half an hour. It is like hot, it takes 1 hour to cool down. So once the milk is cooled down, you will already have a curd at home. The old curd which is already in the form of curd. So see, that 1 spoon of curd, you will be mixing it to the fresh milk which is boiled and already cooled. And what you will do, you will left it overnight or for few hours. Usually we leave it for overnight. It will in the form of perfect form in the thickness of curd. So what is that, how it is happening, that is also of your biotechnology. So whatever this curd, what you tell, curd, you can tell that curd as a inoculum. So what you can tell that curd as a inoculum. So you know that what will be there will be some bacteria which is present in the curd, that is lactobacillus. This lactobacillus will be present in your curd. So when you mix that with a fresh milk, what happens, this lactobacillus will give us a lactic acid. It will have some substance which is lactic acid. This lactic acid will act on the fresh milk and that is converted to your curd. That is converting into a curd, and you can see the the milk is turned into curd. This is the reason how your fresh milk will be turning into a curd. So what happens actually, you can see I told you as lactobacillus, your lactobacteria, okay, from the curd react with the protein assay. I told you lactic acid formed by a product denatures a globular protein and coagulate to produce the solid curd and separates the watery and the protein layer. So what curd, in what you will have it in a curd? In curd you will have lactobacillus, your lactobacteria, that will produce your lactic acid. That lactic acid will help the milk to turn in the formation of curd. Later on you can see a little watery layer and a thick curd present settled in the bottom. So this is also a process of biotechnology in your ancient times. Got it? So now did you understand how your milk is getting converted into curd? You have to remember this, the bacteria and the acid what it is producing. They may ask you, okay. So what is the bacteria present in the curd? Your lactobacillus, okay, lactobacillus bacteria, and that will be giving your lactic acid. So how your milk is converted into your curd. This is your one of the examples. Next, let's move to your next slide. So ancient Egyptian bread making. When was your bread making was done in your ancient Egyptian era? So I know you guys all know how the bread is formed, okay. So what are the first thing you need to make a bread? That is your dough. So what is it? It's nothing but your atta, your wheat atta, some maida. Usually the bread is made in maida, but also you get wheat bread, multigrain bread, and so many types of bread. So now you can call it as a atta. So what this atta is added with the yeast. This atta will be added with the yeast bacteria. So there are so many types of yeast bacteria. So one is Saccharomyces winlocki. Okay, there are Saccharomyces winlocki. There are so many types of yeast. Okay, for this what you will be adding, for this atta you will be adding yeast, and also you can call it as a baker's yeast. What they will be calling that? They are calling as a baker's yeast, because the bakers usually use that, right? So they will be calling as a baker's yeast. So let me give once again. When you have taken an atta and you have mixed yeast, Saccharomyces yeast, this is also called as baker's yeast. From that you are getting it will get fermented, the yeast will get fermented, and you are getting the formation of bread. So what happens? You have taken atta and you mix the yeast in that, and the fermentation is happening, and your bread is formed. That was founded by the ancient Egyptian, in that era actually the bread was found. So next example is your beer making. So you guys know, right, what beer making. So what is this beer? It is a alcohol substances. So what they'll do, your wheat, your cereals, and some of your millets, what they do, they leave it together for a long time. So what happens? They get contaminated, some microbes start reacting on it and everything. So the day by day, so what happened? When they're left aside, when you leave something aside openly, what happen? You get, contaminated, and so many microbes react on that. After, when you just filter that filtration process, you are getting the formation of a beer. That's how your beer is made. That was founded in 5000 BC. Got it? So now you got to know how your ancient Egyptian was. Your bread, which was the bacteria in your curd making, was lactobacillus. Here it's Saccharomyces winlocki. So that is also known as baker's yeast. You can tell that. And how is the beer? You are leaving all the cereals and millets aside for a longer time. It is getting contaminated. It is microbes are acting it. The fermentation is happening. Once you filter all this everything, you are getting the formation of beer. That was happened in 500 BC. Okay, now let's